Hello, my name is Katalina Garcia, and this is my presentation on gonorrhea. So before we go into the presentation, here's a clear objective list that we will be going through, the general information, and then we'll go into gonorrhea in research followed by our references. So starting off, the scientific name for gonorrhea, or the bacteria that causes gonorrhea, is Neisseria gonorrhea. It is a gram-negative STI, and it is one of the most common, reportable, or notifiable communicable disease in the US. As I stated, it is an STI caused by gram-negative Diplococcus Neisseria gonorrhea. So the discovery or the origin of gonorrhea has a lot of history to it. The term of gonorrhea was coined by the Greek physician Galen, which we see pictured here, in AD 150, but the occurrences of gonorrhea goes all the way back to Chinese Emperor Huangdi in 2600 BC, as he mentions it in his textbook. And then we also see it mentioned in the Bible, in the book of Leviticus, as well as by Hippocrates. He called it strangury. So gonorrhea itself has been it's a very ancient disease and has been around for a very long time, but the term gonorrhea was discovered by our Greek physician pictured here. The taxonomy of gonorrhea is shown below. We have the kingdom of bacteria all the way down to the species name that we discussed earlier, Neisseria gonorrhea. From this taxonomy, we see that it is in the phylum of Proteobacteria. This is notable because here we see that it is in the kingdom of bacteria under the Proteobacteria phylum. The Proteobacteria, formerly known as Perbacteria, and its relatives are characterized by a bewildering diversity of morphological and physiological types. Here we see, or just besides rods and cocci, we have curved, spiraled, ring-shaped, appendaged, and filamentous and sheathed bacteria that occur among this phylum. So this is a big varying phylum that we find our Neisseria gonorrhea in. So just to state it, we have our subkingdom Nigibacteria, our phylum Proteobacteria, our class Beta Proteobacteria, and then so on. So going into the structure of our Neisseria gonorrhea bacteria, our bacteria possesses a cell envelope composed of an inner cytoplasmic membrane, and then it has a middle layer of peptidoglycan, and then outer membrane. This outer membrane contains lipoligosaccharides, or LOSs, as well as phospholipid and a variety of proteins that contribute to cell adherence, tissue invasion, and resistance to host defenses. So this bacterial structure is great for attaching onto a host which it needs to survive. So going on, our bacteria and gonorrhea is a naturally competent bacteria with its type 4 pili, which is involved in DNA exchange, specifically proteins, which allows for N. gonorrhea to acquire new genes. This in itself is dangerous in the clinical setting because it has led to a rise in antibiotic-resistant strains of bacteria. Further, N. gonorrhea has surface proteins that bind to receptors on immune cells and suppress the immune response, which results in the host being unable to develop immunological memory. And gonorrhea is also able to invade the immune system through the antigenic variation, and specifically through the structure that we see here. And gonorrhea has a very dynamic polymeric protein filaments that are what I mentioned before, the type 4 pili, which allow for the bacteria to adhere to and to move along surfaces. This is the most notable structure in this bacteria that makes it so dangerous to the human host it attaches itself to. So carrying on, we have the pathogenesis. So during the initial infection, our bacteria adheres to the host epithelial cells through these type 4 pili that we were talking about earlier, which then retract and enable epithelial interactions with other prominent surface structures. So this picture that we see here is a visual representation of this bacteria implementing itself and facilitating transmission. So we can go through the steps here. So we have the pili that adhere to the host epithelial cells. They retract and enable epithelial interactions with other prominent surfaces. Then we see that after the initial adherence, the bacteria replicates and forms microcolonies. Continuing on around step 3, we see that during these initial stages, our bacteria releases fragments of peptidoglycan and lipoligosaccharides, our LOS, and outer membrane vesicles, our OMVs. So these LOS and OMVs then activate the toll-like receptors, our TLRs, and nucleotide binding oligomerization domain containing protein, which we'll abbreviate as our NODs. So these then go and signal epithelial cells, macrophages, and dendritic cells. Continuing on, the NOD and TLR signal for these cells to lead activation of inflammatory transcription factors and the release of cytokines and chemokines. Going on, around step 4, we see that our bacteria releases heptose 1,7-biphosphate, HBP, which then activates our TRAF interacting protein with the FHA domain containing protein. From step 5 to 6, we see the release of pro-inflammatory cytokines and chemokines by these innate immune signaling pathways, which then create cytokine and chemokine gradients that then go on to recruit polymorphonuclear leukocytes or neutrophils to this infection site. Finishing off in step 6 and 7, we see the interaction with and phagocytose of our bacteria, that the influx of neutrophils makes up a purple exudate that then facilitates transmission. So that was just the written response of this visual we see here. And I'm assuming you can see my mouse, but you see how we start here at the initial transmission and then going through, at the initial contact and then going through to transmission. And that is our pathogenesis. So then we have our epidemiology. And as we touched on a little bit earlier, gonorrhea is a global disease. It infects approximately 60 million people annually. In the United States, there are approximately 300,000 cases each year, with 100 cases per 100,000 of the population. In the US, England, Australia, Canada, and nearly all of European countries, rates are often highest among adolescents and young adults, especially those that have multiple partners. These ages are from 18 to 22. Continuing, we see that gonorrhea is disproportionately affecting marginalized populations, these populations being those young adults, those in low income, those in and among sexual and gender minorities, as well as racial ethnic minorities and indigenous communities, as well as sex workers. So that is where we're seeing the brunt of the occurrences. And as we see stated here, approximately 1.6 million new infections occurred in the USA in 2018, half of these occurring in the ages of 15 to 24. So most notably, it is disproportionately affecting marginalized populations, those populations that I did touch on earlier. And continuing on, this is a sexually transmitted infection, so it is spread mostly through sexual contact, but there is prenatal spreading that could be transferred from a mother to the child, specifically affecting the eye as the child is being birthed. So it is generally sexually transmitted, but there is transmission through birth from mother to child. So in the treatment, the bacterial responsible for gonorrhea is, as we've stated before, the Neisseria gonorrhea. It is a highly adaptable pathogen, and its natural competence and plastic genome have contributed to the extensive spread of the antibiotic resistance. In research, what I found and kept finding was that this bacterium is highly adaptable. And as we touched on earlier with its structure, we kind of stated those primary structures that allow for this high adaptability. And so this then results in progressive treatment that needs to be researched, because there's always antimicrobial strains of this bacteria. So we see that this bacteria and gonorrhea progressively develops resistance to antimicrobials that are recommended for treatment, which then results in limited options. But the treatments that are recommended from the CDC is a dual therapy of intramuscular ceftriaxone, as well as in combination with oral azithromycin. And this is the first line of treatment. Continuing, the CDC recommends dual therapy, as I stated earlier, of intramuscular ceftriaxone coupled with the azithromycin. So there are 3 new therapeutics for gonorrhea treatment that are being evaluated, but because of the speed of this bacteria to develop antibiotic resistance, these new drugs are not long-term solutions. So thus far, only 2 gonorrhea vaccines that are used either to kill the whole organism or the purified pili protein, which is essential, an essential bacterial structure that causes the antimicrobial resistance, leading to neither vaccine providing protection against acquiring the disease after immunization. So overall, this disease is highly adaptable, and the current treatment being the combination of the azithromycin and the ceftriaxone. Okay, so as we've touched on before, this is a sexually transmitted disease or sexually transmitted infection. So the main strategies for prevention of this disease are along the same lines of preventing against an STD or STI. So that being stated, some of these ways to prevent STDs or STIs is by contact tracing, condom use, as well as screening. There was research that shows that young adults that have multiple partners are more susceptible of gaining this disease. So it's very important for contact tracing and screening to be a normal process for young adults as they are having multiple partners. This is not to say that someone who's only had one partner couldn't get it, because if it is sexually transmitted and that is not affected by the number of people. But if a certain individual has it, they have it. So continuing on, from Brooks-Vort 2022, we did see that the use of the meningococcal B vaccine can also have cross protection to prevent gonorrhea. What this research found was that there was cross prevention protection for both meningitis B and gonorrhea. So for preventing of this sexually transmitted disease infection, the main prospects are through condom use, they are through screening, but then we also have these vaccines like the meningococcal B vaccine that can also prevent. So gonorrhea and research. The authors of this study that we're talking about, we're looking at the effectiveness of cefixime against ceftriaxone, and that ceftriaxone is what is currently recommended as treatment for gonorrhea. So they used a randomized multicenter study of 124 women and 209 men, comparing 3 single-dose treatments. So the first regimen being 400 milligrams of the cefixime, and then 800 milligrams of that same treatment given orally, or 250 milligrams of ceftriaxone intramuscularly, the ceftriaxone being what is currently recommended in dual therapy for current treatment of gonorrhea. So what the results showed that these researchers found was that there was a 96% cure rate for in result of the 400 milligram dose of cefixime, and then a 98% cure rate for the 800 milligram dose of cefixime, with also a 98% cure rate for the ceftriaxone. So as we can see here in our table here, we could see that researchers concluded that in the treatment of uncomplicated gonorrhea, a single dose of the cefixime, whether it be 400 or 800, given orally, appears to be just as effective as a currently recommended regimen that we did touch on earlier of the ceftriaxone at the 250 milligrams intramuscularly. So this was a very interesting study because, as we stated earlier, the recommended treatment is a dual therapy intramuscular combination of the ceftriaxone as well as the oral azithromycin. And in this research, the authors were looking at the use of single-dose cefixime compared to ceftriaxone, and the cure rates were were very similar. So that's a very interesting aspect as we go to further research. And again, research has to be continually committed for gonorrhea, as stated in this research, because of the prevalence of and the adaptability of gonorrhea. And again, those basic structures, especially the type 4 pili on these gonorrhea bacteria, is leading to the adaptability, which then leads to the recommended treatments being outdated and not long-term, but short-term. So continuing on. This was a lot of information at once. So just to have a quick overview, we did go over the scientific name of gonorrhea, which is the Neisseria gonorrhea. We discovered that Galen, the Greek philosopher, coined the term of gonorrhea. We saw the taxonomy as well as the bacterial structure, the pathogenesis of this bacteria, as well as the epidemiology, and then our treatment and prevention. We ended with the gonorrhea and research, specifically looking at different treatments for gonorrhea that is essential in prevention and treatment because of the adaptability of this spectacularly built bacteria. So here are my references. Page 2. And again, my name is Katalina Garcia. This was my presentation. Thank you.